morning everyone. Today my topic is plasmids. Let's start with the topic. Here is table of content. What we are covering in this topic is that introduction, what plasmids, structure of plasmids, element properties, types, application and application of plasmids. So let's start with the introduction. Joshua Leidenberg in 1952 discovered the term plasmids while working on the conjugation process in bacteria. He also pointed plasmids to refer the transmissible genetic element that were transferred from one bacterial cell to another and determined the malevolence in bacteria. While he found this thing in an E. coli bacteria. Now, what are plasmids? Plasmids are a DNA molecule that separates from N1 and can replicate independently of the chromosomal DNA. Plasmids usually occur naturally in bacteria but are sometimes found in archaea and even in eukaryotic organisms. And plasmids are double-stranded. In many cases, they are circular. The structure of plasmids. Now, molecular biologists have been able to insert custom-built restriction sites into the plasmid so they can be used to learn DNA fragments from genes, and thus they have to propagate those DNA pieces. And plasmids only lead in origin of replication and are useful genes to be considered complete elements. Now, origin of replication. Starting with origin of replication, it is the DNA sequence which directs initiation of the plasmid's application by recruiting bacterial transcription machinery, antibiotic resistant genes. These are one of the important topics where these genes allow the selection of the plasmids containing bacteria by providing a survival advantage to the bacterial host. Multiple cloning sites. This is the short segment containing several restriction enzyme sites enabling easy insertion of foreign DNA. The properties. What are the properties? So they can replicate independently of the bacterial chromosome. They may pick up and transfer certain genes of the bacterial chromosome. They code for their own transfer. And plasmids differ from viruses in following two ways. They do not cause damage to cells and generally are beneficial. They do not have extracellular forms and exist inside cells simply as free as typically circular DNA. Now, types of plasmids. The first one is the F plasmid or effector. Now, also known as the fertility factor responsible for conjugation. It is a conjugated plasmid that contains transfer genes. In addition to transfer genes, it contains several other genetic sequences responsible for the incompatibility, replication, and other functions. Cold plasmids contain genes for the synthesis of bacterioxin known as collagen. These plasmids code for kinase, the proteins that kill sensitive coli cells. It also carries genes that provide immunity to the circular kinase. Now, next one is the R plasmid or R factor. The R plasmid is another group of important plasmid, also known as resistance factor, that carries genes for antibiotic resistance. Some plasmids have only a single resistance, whereas others have as many as 8. These plasmids provide protection against bacteria from both human medicines and from antibiotics produced naturally in the soil. Now, next one is virulence plasmids. These plasmids encode factors to make their host more pathogenic, like the E. coli. Now, metabolic plasmid. This plasmid holds genes for enzymes that degrade the substance known as aromatic compound. Yeah, they are also been helpful in nitrogen fixation bacteria and now replication of plasmid. Mainly due to the replication control system, each plasmid is maintained in the bacterial cell at a characteristic copy number. The replication control of the single copy plasmid is the same as the host bacterial cell, so they can segregate and replicate with the chromosomes of bacteria. In contrast, the replication control of multi-copy plasmids differ from that of bacterial host genome, so that can be undergo more one replication for each replication of their host genome. That is also known as relaxed control. Now, where we can apply them? Unlike viruses, which encase the genetic material in a protective protein coat called capsid, Plasmids are naked DNA. They do not encode genes necessary to encase the genetic material for a transfer of new host. Plasmids are used in genetic engineering to amplify or produce many copies of certain genes. Plasmids are useful in cloning segments of DNA, and also plasmids can be used to replicate proteins, such as a protein that encodes for insulin in large amounts. Well, that's it. Thank you very much for your patience listening.